Sarkar Institute of Pharmacy. Welcome you, sir, for your presentation on insight of clinical research. Over to you, sir. Thank you so much.
Hello. Is it audible, sir?
Yes. Yeah, I'm audible.
Yeah.
Thank you, Anshita madam, for giving this opportunity to me. So first I want to introduce Dilip sir. So a very good afternoon to our respected ma'am, principal ma'am, director sir, my colleagues and dear students. Today is second series of our VTalk series, which is created for sharing the knowledge and experience of various discriminated personalities with us and students and scholars, which may be very helpful to aid in their knowledge and can apply it for policing their career. So today we have one such personality with us who on request get ready to share his knowledge in clinical research. Our today's speaker name is Dr. Dilip K. Raja sir, and who is scientist C in the Division of Epidemiology and Communicable Disease at Indian Council of Medical Research (ICMR), New Delhi. And sir has done his PhD from Acharya Nagarjuna University at Andhra Pradesh with a specialization in pharmacology. And sir have very good industrial experience in preclinical as well as clinical research. Sir previously worked in Central Drug Standard Control Organization, that is CDSCO, that is at New Delhi. He also worked as drug safety associate in Quintiles, presently we are known as IQVIA at Bangalore East. Also, he worked as a research associate in pharmacology laboratory for clinical research at Lella Nutraceuticals at Andhra Pradesh. And also, he has teaching experience and worked as assistant professor in Nirmala College of Pharmacy as well as Bharat Institute of Technology. And sir has huge hand-on experience in clinical as well as clinical research. And today, sir will share the various aspects of clinical research. So I am again welcoming and requesting to Dr. Dilip sir, please share the knowledge on the topic of insights of clinical research. So sir, please proceed.
Yeah. Respected Dr. Deep Kaur sir, the principal of the ma'am, sorry, the principal of Rawatpura College of Pharmacy, and the management of the Rawatpura College of Pharmacy. First of all, thank you for inviting me as a guest speaker to give a lecture on the insights of the clinical research. And thank you for your warm welcome. And I'm very honored to present this particular topic to the students as well as the delegates. Shall I share the screen, sir? Give me a minute. I'm just sharing the screen. Is it visible, sir?
Yeah, your screen is visible, sir.
Yeah. So today I'm going to give a lecture on insights of clinical research. My contents are the new drug development process, clinical research, clinical trials and types of clinical trials, stakeholders of clinical trials, phases in clinical trials, and the regulatory body which regulates the clinical trials, an overview of clinical trials and the safety reporting involved in the clinical trials, and the completion or termination of the trial and opportunities in the clinical trial. So these are the different topics I'm going to discuss in the short while. So you must have aware of the new drug development process. So what are the different phases involved in the new drug development process? So it starts from the target selection. Target selection. So what is a target? So let's see, like HIV, AIDS. What is the target for AIDS? The HIV is a target. And what is the human immunovirus? And what is the target for the tuberculosis? It's a mycobacterium. It is a target. So we will first select the target of the disease to which we have to prepare or to develop a drug, and then we'll get to the next the step of the drug development process, which is target validation. And then we'll get into the the drug selection or the lead selection. So in this, like will begin to collect the bulk of the the drugs from the different sources, from animal source, from the plant source, as well as from the chemical entities. And then we will begin to select through the in vitro process, and then using different kinds of techniques, high throughput screening and all. So we will begin to select one particular drug or a lead. We will call it as a lead, and then we will optimize the lead using the docking techniques, the computer aided drug design. And then after once the lead optimization is done, we will get into the preclinical testing. Preclinical testing means animal testing, means so once the lead is optimized or the drug is optimized, we can't directly go and directly give it to the humans. So that is the reason why we will first see in the animals whether the efficacy and the safety parameters are good or not. And then if the drug passes the preclinical testing, then we will get into the clinical testing, which means the trials on the humans. So we will do the research on the humans. So we will have the overall idea, we'll have the whole big picture of the new drug development in this particular slide. So if you see the discovery phase and the candidate profiling phase, in these two phases, we will begin to select a proper lead or the proper, the optimal, the drug. So we will select a drug in this particular two processes, and then in the next phase, in the preclinical trials, we will begin to do the research on the animals. So we are going to test on the animals. So that particular phase, we will call it as preclinical trials. So if a drug, the lead compound, if it passes the preclinical trial, means if it passes the animal testing, if it is showing the efficacy as well as if it is showing the safety in the animal studies, then we will get into the clinical trials, which is the phase one of the clinical trials. But before getting in the clinical trial, we have to get approval from the regulatory body. So we have to file an application called investigational new drug application. So if you see here, so the IND means investigational new drug application. So we have to apply to the concerned regulatory body for the investigational new drug application. Sorry, investigational new drug. So that particular application, we will call it as IND application or investigational new drug application. So after the scrutiny of the documents, we have to submit the whole data right from the lead selection, to the preclinical trials. So we have to submit the whole data to the regulatory authority. Then they will scrutiny the whole documents, and then if they are convinced, then they will approve the IND. Means they'll approve to do the trials on the humans in the clinical trial phase one. So clinical trial phase one and phase two and phase three. See, it goes on, and before ever getting into the phase three, again we have to apply for the new drug application form. Investigational new drug application. Now it is a new drug application. So this particular new drug application should be applied to the regulatory body in order to carry out the phase 3 trial. So we are going to learn about all the phase 1, phase 2, phase 3, and phase 4, right? So this is all it goes. So next thing, let me explain the clinical research. So the clinical research is a branch of healthcare science that determines the safety and effectiveness of medications, devices, diagnostic products, and treatment regimens intended for the human use. So clinical research is different from the clinical practice. Don't get confused with the clinical practice, with the clinical research, with the clinical practice. Because in the clinical practice, we are using the established treatments. But in the clinical research, we have to collect the evidence to establish a treatment. Means we are doing some research to establish a treatment protocol is called a clinical research. Using the existing clinical or using the established the treatment is called the clinical practice. And then the clinical trials. So clinical trials in relation to a new drug or the investigational new drug, it means the clinical trials, any systemic study of such new drug or investigational new drug in human subject to generate data for discovering or verifying its clinical effects or pharmacological effects, including pharmacodynamics and pharmacokinetics and adverse effects. So we are discovering, we are, doing some research on humans for discovering or verifying its clinical effect, means the action of the or the effectiveness of the drug, the pharmacological activity including pharmacodynamics and pharmacokinetics. Pharmacodynamics and kinetics you must be knowing, and the safety of that particular drug, means the adverse effects of that particular drug. So this particular definition is according to the new drugs and clinical trial rules 2019. So we have different types of clinical trials. So based on the researches we have observational, based on the investigators, decision. We have the observational study as well as the interventional study. So what is observational and what is interventional? Observational study does not involve any kind of drug administration to the humans, means as I said you, we have already discussed that clinical trials is nothing but research on the humans or trials on the humans. Now, if we give any kind of drug, we will call it as a interventional study. And if we, without giving any kind of experimental drug, only observational, if we do only observations, we will call it as a observational study. So we have observational study as well as the interventional study. And here the second category is based on the purpose. We have the preventive trials, means the vaccine trials, screening trials, diagnostic trials to to establish the new kind of diagnostic tools and diagnostic kits, and the treatment trials, quality of life, means to increase the quality of life, and the genetic trials to find out which kind of gene is responsible for the disease, and which kind of gene we have to target to cure the disease. And then compassionate use of trials, means solitary trials, we'll call it as solitary trials. If we don't have any kind of treatment for the established treatment, we will use this compassionate trials. And epidemiological trials means this is the epidemiology, means we, so epidemiology means you must be knowing that. So in what kind of population this particular drug is acting, and what kind of population drug is not acting, and the what kind of population is the disease is more prevalent, and in what kind of population disease is not more prevalent. So this kind of, we have different types of clinical trials. So these are the different kinds of clinical trials. Now we will see the stakeholders of the clinical trials. Who are all the persons, or who are all the stakeholders involved in the clinical trials? So we have a sponsor, contract research organization, which is also called as CRO, principal investigator, ethics committee, regulatory authority, and the study subject, who means the study participants or the volunteers. So we are going to learn, each and everything in a brief. So we are very short of time. That is the reason why I did not keep the individual slide for each and everything. So I'll explain you each and everything. Sponsor. Who is a sponsor? The sponsor is the one, who sponsors the trials, or who gives the capital for the trial, who funds the trial. So sponsor may be of the private organization or the government organization. Private organization means sponsors like the industries and the companies like Johnson and Johnson, Pfizer, and Glenmark, Dr. Reddy's. These, the private companies, the whoever are funding the particular trial. And the other kind of sponsors are the government organizations. Government organizations like ICMR, Indian Council of Medical Research, DST, Department of Science and Technology, DPT, Department of Biotechnology, and then AICT. So like these are all the different, whatever the organization which funds, which gives capital to carry out a research, we will call them as a sponsor. We will call them as a sponsor. The second one is contract research organization. CRO CRO. What is a CRO in the clinical trial? So the name itself, we can say that contract research organization. This also do the research, but on contract it does. So what is a contract? So why this contract research organization is important in the clinical trial? Anyway, the sponsor is sponsoring or the funding the trial and can do whatever the CRO can do. But the sponsor will begin to hire the CROs, contract research organizations, to increase the transparency of the data. Means like if okay, I'm going to explain after the principal investigator, ethics committee, and regulatory authority about the contract research organization, because you'll have a better idea if I explain after this. So now the principal investigator, I'll tell you the principal investigator. You know, principal investigator is one who does the real time research, means in the site. Like as I said, your sponsor is the one which gives the money, and the principal investigator is the one who does the research on the human volunteers. Means he has the one, he is the one who is going to recruit the volunteers. He is the one who is going to look after all the real time research. So principal investigator. And ethics committee, ethics committee is the one. Why like okay then, like we'll have a better idea. Like just remember these names: sponsor, CRO, principal investigator, ethics committee, regulatory authority, and study subjects. Anyway, I'm going to explain in the coming slides. Now, the phase. What are the different phases in the clinical trials? So we have total of 5 phases: phase 0, phase 1, phase 2, phase 3, and phase 4. Right? So now let me explain about each and every phase. So we have the phase 0. So now let's assume a drug just passed the preclinical research, means just passed the animal studies. Now it is about to get into the clinical trials, means it is about to be tested on the humans. So let's say it got the approval of the regulatory body by after applying the investigational new drug application. Once the drug gets the approval from the regulatory body, then it gets into the phase 0. In this phase 0, we will give microdosing. So only we will take the 10 people, 10 to 20 people. So very small number of people we will take, and we'll give the microdosing. We'll give in the nanograms or the micrograms. So we'll give the microdosing, and then we will see the pharmacokinetics. What is the pharmacokinetics? See the ADME. What is absorption rate? What is the metabolism? What is the distribution? And what is excretion? Like what are these 4 parameters? We will begin to see the pharmacokinetics, particularly see the bioavailability and the half-life of the drug in this particular phase 0. So normally clinical researchers will begin to carry out this particular this particular phase. So phase 0 is the one which is carried out in the 10 or the 20 people, means a small number of people, small number of participants. And in this particular phase 0, we are going to see the pharmacokinetics as well as the bioavailability of the drug. So bioavailability of the drug, you must be knowing the rate and extent of the drug absorption into the body, into the blood circulation, right? And then if it passes, if the everything is well, if it passes the phase 0, if the drug has got the good pharmacokinetics, absorption is good, distribution is good, metabolism is also good, and excretion is also good, then it goes into the phase 1. Means phase 1, phase 0 and phase 1 both, we will carry out in the healthy volunteers. We will not take any kind of patients. We will take healthy volunteers. In this phase 1, we will take 20 to 100 healthy volunteers. Normally we will take healthy volunteers, but in some cases, in the case of cancer drugs, we will take cancer patients, because we can't simply give it to the normal patients. We can't simply give the cancer drugs to the normal patients. So Uh, phase 1 is carried out in a small number of participants, but little bit more number when compared to the phase 0. So we will see the dose ranging and safety, especially safety. Almost it is a similar, it is extension study of the phase 0. Phase 1 is a kind of extension study of the phase 0. So here also we will see the pharmacokinetics as well as the safety of the drug. Here we will see the subtherapeutic and the subtherapeutic, like sometimes in a very few cases we will see whether in the case of cancer and in the case of few studies we will see the pharmacodynamics of the study also in the phase 1, but normally we will see the pharmacokinetics of the drug in the phase 1. The next phase, like if it is a safe, means if like drug, that lead molecules which came, like that particular lead molecule which came from the preclinical research, which came from the animal research, otherwise safe, so it can be rejected in any phase. So it has to pass phase 1. If it does not pass, it gets rejected. So we will not do phase 1 study, right? So it has to pass the phase 0. Only then we will perform phase 1 studies. Again, it has to pass the phase 1 studies. It has to show the safety. Only then we will get into the, we will carry out and go a step forward into the phase 2 studies. Or else it gets rejected, we will stop the study there itself. So once the drug passes the safety test and gets into the phase 2, the phase 2 is the exploratory study, which we will, in this particular phase 2, we will take 100 to 300 participants and we will see the actual pharmacodynamics of the drug. Means we will take the patients. In the first two phases we took the healthy volunteers. Now in the phase 2 we will take the patients which are suffering with a particular disease. So if we are doing a research on the diabetes, we'll take the diabetic patients. If we are doing research on cancer, we'll take the cancer patients. If we are doing research on the AIDS, we'll take the AIDS patients. So in this phase 3 we will take, so in this phase 2 we will take the patients. So 100 to 300 patients we will recruit. We will make them to enroll into the study, and we will see the efficacy of the study, efficacy of the drug, as well as the side effects of the drug. Means along with the pharmacodynamics we will also see the safety of the drug. So safety here in the phase 1, the actually we will see the pharmacokinetics, but in a little, in a little concern we will have, like we will see the pharmacodynamics. In the phase 2, we'll see the majority, we will see the efficacy of the drug, but in the little portion, we also have the safety concerns. So we will also see the safety concern of the drug in the phase 2. So if the drug passes the phase 2 also, like if it establishes it is actually working, if the drug is actually working in the phase 2 in the patients, then the drug passes and it crosses and gets into a next phase called the phase 3, right, which is the multi-center trial. So phase 2 we will otherwise call it as exploratory trial, and the phase 3 is the confirmatory trial. Means in the phase 2 we have established the efficacy of the drug, means it is actually acting. We are seeing the whether the drug is acting or the showing action on the disease or not. Means whether the drug is decreasing the or treating the disease or not. Means like let's say diabetes, if you take if the drug is supposed to have, the reported to have the anti-diabetic activity, whether the diabetes means the sugar levels are decreasing or not, we will see that in the phase 2. Once the drug is acting well, means once the drug is showing some activity, then the drug is going to get into the phase 3 studies called the multi-centered trial, multicentric trial. In this phase 3, we have like the huge number of participants, around 3,300 to 3,000 participants across the globe, across the different populations, different ethnicities, and different races. Let's say the single drug, now let's say one drug is acting in India, the population in India, and it is established in the phase 2. Now, like it is not enough, right? So we have to like confirm the activity in different locations across the globe. So we have to see that particular drug in the Africans, in the Americans, in the Australian people, in the in the people who are living in the Europe. So we'll see in the different ethnicities, different kinds of populations, and the different kinds of races across the globe. So this is the phase 3 to confirm. We are expecting the same the activity in the phase 3 also in the different populations also. So this is all about the phase 3. If the drug shows the some positive action in the phase 3 also, so we can directly launch it into the market, but we have to apply for the new drug application, right, to the to the concerned regulatory body. And right from the beginning, I was speaking about the regulatory body. Like I'll tell you what is regulatory body in the coming slides. So we have to apply the new drug application to the regulatory body, stating that uh, our drug is passed in all the three phases, and right from the the lead discovery to the phase 3, will submit all the data to the regulatory body. And the drug if the regulatory body convinces with the data, and like if they're satisfied with the data, then they'll give the approval to launch that particular drug into the market. But the drug may fail in any of the phases, in the phase 0, phase 1, phase 2, and phase 3. Any of the phases, it can fail. So now let's imagine the drug passes the phase 3 also, and it gets launched into the market. Now comes the phase 4, which is called as post-marketing surveillance. So post-marketing surveillance is the phase which happens after the phase 3, means which happens after the drug launching into the market. So what is this post-marketing surveillance? So we have seen the phase 0, phase 1, phase 2, and phase 3, and we have seen the safety in all the three phases, but we can't see the adverse events or the side effects of the drug in the long term. Like let's say phase 0 is 6 months, phase 1 is 6 months, phase 2 is 1 year, phase 3 is 3 or 4 years. We can see maximum 3 years of time. We can see the side effects of the particular drug in a maximum 3 years of time. But what about the long-term effects of the drug, like 7 years, 8 years, and 10 years? So we will see the long-term side effects or the long-term adverse effects of the drug in the post-marketing surveillance. So this particular phase 4 is called post-marketing surveillance. And many of you people got confused with the pharmacovigilance and the post-marketing surveillance, because many of them think that post-marketing surveillance is nothing but the pharmacovigilance. It is equal to pharmacovigilance. But pharmacovigilance is different and post-marketing surveillance is different. Though post-marketing surveillance is one of the part in the pharmacovigilance, but it is not the whole pharmacovigilance, right? So this is all about the phases of the clinical trials. Now comes the regulatory body. Right from the beginning, as I said, you regulatory body, I was mentioning about the regulatory body. I said you, like in the phase 1, in the phase 0, phase 1, in the whole of the clinical trial, simply we can recruit the volunteers, like recruit the volunteers and do the research directly on the humans. Like, who is the one who overall supervises the study? Right? Like, let's say the alcohol. Let's imagine the alcohol. We have the the excise department to regulate the alcohol, right? Similarly, we have the regulatory body in each and every country. Each and every country have their own regulatory bodies to look after all the the clinical trials and the research and the drug related imports, exports and all. So who is a regulatory body in India? So Drug Controller General of India, Drug Controller DCGI. So he's under the Central Drug Standard Control Organization, CDSCO. So Central Drug Standard Control Organization, CDSCO, is a regulatory body of India, and in that the like uh, the highest the position is a DCGI, Drug Controller General of India. He is the one who has got the prime responsibility for regulating the clinical trials in India, right? So we have new drugs and clinical trials rules 2019 under the Drugs and Cosmetics Act 1940. So earlier it was Schedule Y. So just remember and note it down this thing. Earlier, we have all the requirements and the guidelines for the clinical trials in the Schedule Y of Drugs and Cosmetics Act, but now it has changed to New Drugs and Clinical Trials Rules 2019 with effect from March 2019. So you have to note it down, this particular updation. Earlier we have Schedule Y of Drugs and Cosmetics Act. Now it is New Drugs and Clinical Trial Rules 2019. It came into the effect from March 2019. So this is all about the regulatory body. Now, the overall view of the clinical trials. So we have earlier we have discussed about the stakeholders of the clinical trials, the sponsors, the principal investigators, and all. So all those stakeholders I'm going to explain in this particular site. Now the study design and the study documents, right? The study design. Initially, whenever the sponsor wants to initiate a study, they have to sit down and they have to write a protocol. They have to prepare a protocol for the study. They have to design a study, right? And then after designing the protocol of the study, they have to submit it to the regulatory authority, which is a CDSCO. So they have to submit all the documents. So here, study documentation in the second, the box you have in the flow chart, you have a study documentation. The study documents right from the beginning of the journey, like in the sense journey of the lead molecule. So the lead molecules, initially we have the bulk of lead molecules. We have finalized the lead molecule and we have carried out a research in the preclinical. It means in animals, and then all the data we have to submit it to the DCGI for the approval of the clinical trials, to carry out the clinical trials. Now, so this regulatory body, once the sponsor, so-called sponsor, whether a government organization or the private organization, private companies, once they submit the whole documents to the regulatory body, which is CDSCO in India, they will scrutinize the documents and they will see whether the safety protocols are followed or not, whether the drug is safe or not. And if they are convinced and if they are satisfied with the data which they are submitted by the sponsor, then they will issue an certificate, no objection certificate, called CTNOC, Clinical Trial No Objection Certificate. Kindly note it down, these particular documents. I could not able to capture in the PPT because of the lack of time and the lack of the space also. CTNOC, the Clinical Trial No Objection Certificate, is being issued by the regulatory body, which is CDSCO, to the sponsor. Means it is saying that we don't have any objection to carry out this particular trial. So once they get the CTNOC from the regulatory authority, which is CDSCO, then they'll start searching for the investigator selection. Who is an investigator? As I said to the sponsor, if let's imagine if it is if a phase 2 or phase 3 trial, the sponsor is the one which gives you money, as I said you, the money as well as the study design. And then the investigator is the one. Normally investigators are all the doctors. So who are already established in their particular fields, if they want to do research on tuberculosis, they will find the doctors who are good in the tuberculosis, the pulmonologists. If that particular sponsor wants to do some diabetes study, they'll go and see the endocrinologist or a diabetologist. If they want to do a research on the cardiology, they'll search for the established cardiologist. So investigative selection will takes place after getting CT and OC from the regulatory authority. So investigators are normally doctors who are already established in their particular fields. So once the investigators were selected, and the places where the trials are going to perform were called as investigation sites or clinical trial sites, means normally hospitals. Normally hospitals means the uh, investigators are the doctors, and the investigation sites are the hospitals, right? So in each and every hospital where the clinical trial is going on, because why the hospitals? Because if we want to do a trial on the patients, so we need hospitals, because patients will come to the hospitals only. If we want to do some research on the cardiology, like in the cardiac diseases, we want some cardiac patients. So cardiac patients normally available in the the hospitals, the specialized hospitals who where the cardiologists are present, right? So in each and every hospital or each and every site, we will have a ethical committee. One ethical committee has been designed, like there has been constitute in each and every clinical trial site. So in this particular ethical committee, it constitutes of one social worker and one lawyer, one scientist outside the institution, one scientist inside the institution. So we'll have, like, you might have heard about the CPCSA ethics committee. Similarly, we have the human ethics committee, which is constituted for each and every hospital, each and every site. So this ethics committee will begin to monitors and will begin to supervises the whole clinical trial process in that particular site. So it all it might most of the time it works on behalf of the patient, because the subjects who are going to enroll should not get exploited by the hospitals or by the sponsors. Now, and we have right. So once the study is being initiated, once the investigator is selected and the study site was selected and the ethics committee was selected, ethics committee should be the constituted members should notify to the regulatory body again, right? So once all these are set ready, now we have to start enrolling the patients, enrolling the subjects, right? Because we have to do perform like subject means here the people who are and like involved in the clinical trials. We will call them as subjects or participants. So once the like before ever the participation means enrollment of the participants happens, we have to take the consent from that particular participant, that particular form. We will call it as informed consent form, ICF. This also very important. Like students who are listening, you can note it down. Informed consent form is mandatory before the enrollment of the subject. So this is nothing but the consent we are taking from the participant to carry out the research on his body, right? So this particular informed consent form, like it is not simply a document which is to be signed by the participant. It also includes audio and video proof. Means while the participant is signing on the informed consent form, the audio as well as the video, means the terms and conditions are to be explained by the that particular the staff member in that hospital to the subject, or or by the doctor to the subject. So that will be recorded, audio as well as the video will be recorded. So that is a proof that the subject, came and signed the informed consent form, means he is giving the consent wholeheartedly without any force, because we are also, recording the audio and video evidences, like we have also audio and video evidences in the future. So once the informed consent form is done, the consent process is done, the subject will be screened for this, like if he is suitable for the trial, he's been enrolled into the study, and then the study will begin to start normally. Like the we will give the, if it is an interventional study, we will give the experimental drug to the human, and we will see the outcomes of the particular study. Now, the fine. In between, we have a safety reporting. What happens, like in between the study? Like, let's imagine a participant gets enrolled, and after enrollment and after the study initiation, means after the study IP, means investigational drug administration. If any kind of adverse event happens to that person, or any serious adverse event happens to that person, what happens? What where we should report and what we should do. So this particular process we will call it as safety reporting or pharmacovigilance, as I said you earlier. I'm going to explain this particular pharmacovigilance in the later slide. Just hold on. So this is all about the clinical trial, and now during the clinical trial, what is the role of CRO here? So she's the one who has been hired by the sponsor, means sponsor is the one which gives money to the CRO, which hires the CRO to monitor the trial. Means, see, sponsor anyway, it is doing its work, it monitors the trial, but along with it, it gives to the third party, it gives, the contract to the third party to monitor the trial, to maintain the transparency, because the data, the monitoring, it starts from the, compliance of the GCP. GCP means good clinical practice. So they have to follow the GCP. India, we have Indian GCP. I could not able to capture in the PPT enough. So every investigator, every sponsor, every ethics committee, everyone have to, follow the GCP rules here. If there is any incompliance of the non-compliance of the GCP rules, or non-compliance of the regulatory, the requirements, non-compliance of the, deviation of the protocols, or non-compliance of any kind of protocol deviations, any kind of, malpractices in the sites, will be monitored by the CRO, and then report to the sponsor. So just to increase the transparency of the data, and just to increase the, surveillance of the data, surveillance of the site as well as the trial. So we will, the sponsors normally recruit the CROs, contract research organizations. So finally, at the end of the trial, the data will be generated. Data means, like whether the how many subjects gets enrolled, how many participants got enrolled, and how many participants the IP, means investigational drug, shows efficacy, and how many subjects it doesn't show efficacy. We will call it as a data. So the whole data will be gathered and submitted to the sponsor as well as to the CRO. And now both of the parties will begin to analyze it using some statistics, many statistical softwares. Like in preclinical, normally we will use GraphPad, Prism, Origin, but in a clinical trials, normally we will use SAS, statistical analysis software. So these SAS programmers will be recruited by the contract research organization as well as by the sponsors. So both of them will begin to perform the statistical analysis, and then they'll finally submit it to the sponsor. So by this, the study will begin to end. The closeout of the clinical trial happens. So now comes the safety reporting. So safety reporting, as I said, your pharmacovigilance. Pharmacovigilance is also known as drug safety, is a pharmacological science relating to the collection, detection, assessment, monitoring, and prevention of adverse effect with pharmaceutical products. Now, whenever we speak about the pharmacovigilance, we have to speak about the two terms called adverse events and adverse drug reaction. So, adverse event is nothing but any untoward medical condition or occurrence in a patient or clinical investigation subject administered a pharmaceutical product. As uh, I said you in the clinical trial will give the pharmaceutical ingredient or the IP investigational drug to the human or the clinical subject, right? In that particular time, if any untoward occurrence happens in that particular period, then we will call it as an adverse event. So that particular adverse event may or may not have the causal relationship with the drug, means may or may not related to the drug. It may be related or it may not be related to the drug which you are giving. Let's say a patient is he's having the medical history of the epilepsy. He's already having epilepsy. Right? Now we don't know that the patient is having the epilepsy. Now once we give the drug, investigational drug, after one day if the patient gets the seizure, the episode of the seizure, so we don't know whether it is because of the drug or not, but it is an event, right? One event happened. It is not necessarily because of the drug. It may be because of the medical history of that particular subject also, because he's already having epilepsy. So if we have a doubt, if it is not clear, then we will call it as a adverse event. It is an adverse event which is not necessarily have the causal relationship with the drug. And if it is, if it is, have the if the same particular event, if it is have the causal relationship with the drug, we will call it as an adverse drug reaction, ADR. We will call it as ADR. So if the drug have, if the event doesn't have the causal relationship with the investigational drug, we will call it as adverse event. If the event have the causal relationship with the drug, we will call it as adverse drug reaction. So, and now adverse drug reaction, simply adverse drug reaction, uh, like uh, adverse drug reaction or adverse event. Again, we will categorize the adverse event into serious and non-serious. So all adverse event cannot be as serious adverse events. Some are normal adverse events, some are serious events. So we have certain criteria for the serious adverse events. So these are the 7 criteria. One is death. Second one is life-threatening. Third one is hospitalization. Fourth one is permanent disability. Fifth one is congenital anomaly. Congenital anomaly means birth defect. And fifth one is the permanent damage, require intervention to prevent permanent impairment or damage, and important medical event. So if that particular adverse event falls into this particular 7 criteria, we will call it as the serious adverse event, SAE, serious adverse event. So you have to basically understand that whenever there is an serious adverse event happened in the trial to the subject, it will be reported to the regulatory authority immediately by the sponsor, by the investigator, and by the ethics committee also, right? So I tell you the timelines of reporting of essays, and you also should understand that AEs need not to report to the regulatory authority. Means all adverse event. For example, small at the injection site, small inflammation. Whenever you have a vaccination done, you will have a small inflammation or small you know the arrhythmia the forms at the site of injection. It will subside within 1 hour or 2 hour. It will not be reported to the regulatory authority. But what needs to be reported if that particular arrhythmia requires the hospitalization of that particular subject, means because of that particular arrhythmia it becomes too much high, and the patient was hospitalized because of arrhythmia, then erythema, then that particular that particular event should be reported to the regulatory authority, which is CDSCO. So we have certain timelines here. If any SAE happens in the site investigational site, the investigator has to report to the regulatory authority, to the sponsor, as well as to the ethics committee within 24 hours. These timelines are very important. Kindly note down the students who are interested to get into the clinical research as a career option. Uh, Investigator's timeline is at 24 hours. Within 24 hours, he has to report to the regulatory authority, sponsor, as well as to the ethics. All the three stakeholders, the investigator has to report. And the sponsor means the investigator, anyway, investigator is reporting to the sponsor, right? The sponsor has to report to the regulatory authority within 14 days. Within 14 days, we will call it as a 14 days report. And the ethics committee, as I said, the investigator initially he reports to all the three stakeholders along with the CDSCO. The sponsor again has to report to the CDSCO with the causality assessment. Means the sponsor should have a medical monitor, means medical reviewer. He has to review the event and then he has to give the causality assessment whether it is related, means the event is whether the event is related to the drug or not, investigational drug or not. And the ethics committee also, it has to review the whole event, and then it has to submit to the regulatory authority within the 30 days of time. So the regulatory authority will begin to receive the SAE report from all the three stakeholders, which is PI within 24 hours, sponsor within the 14 days, and the ethics within the 30 days. Now, as I said, the investigator will be reporting the SAE within 24 hours, right? But he has to regularly submit the follow-ups till the subject recovered from the SAE. If it is a death, no need of submission. But if it is an injury, like if it is a fever, let's say he got admitted in the hospital for 5 days. After 5 days, the final SAE report also should be submitted to the regulatory authority, to the sponsor, as well as to the ethics committee. Again, these sponsor and ethics committee should report to the regulatory body, right? Now, uh, let's say if the SAE is death, means the subject died in the clinical trial, and if the death is because of the drug, it is related to the drug, because of the investigational drug the subject died. So what is the consequence? The subject died, he has to pay the compensation for that particular death, right? So this particular compensation will be ordered by the three reports from the three stakeholders. Once it reaches to the regulatory authority will begin to scrutinize all the three stakeholders causality assessment, and then they'll begin to, if the drug is really related to the investigational drug, if the event is really related to the investigational drug, they will order a sponsor to pay the compensation amount to the participant. Like if the patient died, that particular compensation will be paid to the family members. If the patient hospitalized, that particular medical expenses as well as the compensation will be paid to that particular participant, right? So how this compensation was calculated? If it is a death, the compensation will be calculated by using this particular formula, B into F into R by 99.37. So B is a base amount, which is 8 lakhs. 8 lakhs is a base amount, and F is a factor depending on the age of the trial subject as per the Annexure 1. Annexure 1, it is mentioned in the new drugs and clinical trials rules 2019. If you can able to check the new CT rules in the internet, you can check, you will find the Annexure 1. And then R equal to risk factor depending on the seriousness, means F means, if the subject's age is too high, the compensation will begin to become less. If the subject age is too young, if he's too young, the compensation will more. So that is a factor. The factor like F is like if the age is more, the F is less. If the age is less, the F is more. And R is the risk factor. Risk factor depending on the seriousness and severity of the disease and the presence of comorbidity and the duration of the disease of the trial subject at the time of enrollment in the clinical trial between the scale of 0.5 to 4 means 0.5 is terminally ill patient means very ill patient, and 1 is a patient with a high risk, 2 is a patient with moderate risk, 3 is patient with a mild risk, and 4 is the healthy volunteer. This is the calculation for the compensation of the death, and this is the compensation in the permanent disability means C into D into 90 by 100 into 100. So where D is a percentage disability of the trial subject which has suffered, that disability will be decided by the committee in the CDSCO, and the C is quantum of compensation which would have been due for the payment for the trial subjects normally in the case of death of the trial subject means like this is a quantum of compensation in the case of death means permanent disability means it cannot be reversed, right? So in the case of death, how much is the compensation normally is given? That particular compensation will be is nothing but the C, right? And if it is an injury, the minimum wage per day of the unskilled labor in the unskilled worker in Delhi multiplied with the number of days of hospitalization into 2. This is the compensation in injury. So these are all the compensation calculations of the death, permanent disability, as well as the injury. In case if the drug, in case like if the event is related to the drug, the serious adverse event is related to the investigational drug. So and then the completion, finally the safety reporting is also over, and if the subject out, if the trial the goal of that particular trial or the outcome or the objective of that particular trial is achieved, then the completion of the trial will happen, the ensuring the outcomes achieved, or if the efficacy is established, then the trial will begin to complete. And in case if there are more serious adverse events happened in between the trial, then they'll terminate the study in between. If or else if they fail to establish the efficacy, like these are the two concerns. One is the safety concern, and the other one is if the if the particular drug is fail to establish the efficacy, then the particular trial will be terminated in between. So keeping the students in upcoming budding pharmacists in the mind, I just made one more slide, the opportunities in clinical research. So, because many of the students might be interested in getting into the clinical research. So these are all the different career, the options available for the budding pharmacist. One is the clinical research coordinator, CRCs. I said you investigator, principal investigator, right? And investigator works in the clinical trial site. In the site, this particular CRC means CRC will present, clinical research coordinator will be working along with the principal investigator in the clinical trial site. He'll be assisting the principal investigator. And next one is CRA, clinical research associate. So I said you clinical research is a contract research organization. I'm sorry. I said you contract research organization, right? CRAs will be working along with the sponsors as well as contract research organizations to come and monitor the sites. So CRA, Clinical Research Associate, is the one who works with the sponsor as well as the clinical research contract research organizations to come and monitor the site for the well-being and the compliance of the study with the GCP or new city rules. And the drug safety team is involves in the safety reporting, right? Pharmacovigilance. Again, I'm sorry. Drug safety team will have their own hierarchy. manager, first line manager, second line manager, associate, or specialist. These kind of team will be there for the drug safety team, and the regulatory team. So I said you about NDA, INDA, CTNOC. So all these kinds of regulatory communications will be taken care of the regulatory team, and the project management team will be working along with the sponsor to manage the whole project across different clinical sites. And the statistical programming team is a team which works to analyze the data which gets from the clinical study. Like in the study, once a study completes, you'll get the data. That particular data will be programmed statistically using the statistical programming team. Normally SAS team will be working along with it, and the QA team also works with the sponsor as well as the CRO to clinical assurance, to assure the quality of the QA means quality assurance, right? To assure the quality of that particular study. And the medical writing team, it starts from the writing of the protocol of the study as well as the final conclusions. All these writing part will be taken care of the medical writing team, and there also we have their own they have their own hierarchy, right from the manager to the associates and all. So these are all the different. Maybe I might have missed one or two in a you can also search in the Google. So these are all the best, with the best of my knowledge. I could able to collect this particular the career opportunities for the budding pharmacists. So these are all the this is all about the insights of clinical research and also the opportunities in clinical research. And if you have any doubts, you can ask me.
If students or scholars have any doubts, you can ask. So you can see the chat box, so their students will be typing. I'm asking that, sir. We talk about the very safety and efficacy of new drug applications in the clinical trial, but shall I repeat?
Yeah, ma'am. Actually, can you please repeat? I could not.
So we talk about safety and efficacy of new drug application in clinical trials, but in
Yeah.
this pandemic, vaccines are prepared in very short span of time. So within
Yeah,
six
yeah,
months, it is taking 1 year to come into market. Sir, how
yeah, yeah.
safety and efficacy will be accomplished of these vaccines, sir?
Yeah, exactly. If you see my presentation, we have, I said you about the different types of clinical trials. In that, compassionate use trials. If you see
Okay.
the slide, right? So, compassionate use trials means like this is a kind of solitary trials. Like if we have no treatment options available in the market, and especially kind of pandemic happens, we don't have any options available in the market. We don't have treatment options, we don't have any cure options. So in this kind of situations, we will go for compassionate use trials, means solitary trials. In these trials, like will we won't see the efficacy and the safety of the drug that much, like controlled interventional trials. We don't see the safety because that is an emergency, that is an health emergency, right? So in those cases, we will use this compassionate trials, ma'am. So in that case, much of the efficacy and the safety we will not see. But during these trials, if any of the safety concerns arises, then immediately we will stop the trial.
Okay, sir.
So that is the issue. This in the emergency grounds, in emergency cases like of pandemics and all, we will use this kind of clinical trial, ma'am.
Okay, sir. Thank you,
Yes,
sir.
you're welcome, ma'am.
Yeah, Dilip sir,
Yeah,
one more question I have.
yeah, yes, sir.
Suppose if we have completed our preclinical studies
Yes, yes, sir.
and we want to go for clinical trials,
Yes, sir.
and so first we have to take ethical permission from, as you know, human ethics committee, that is from hospital, or first we have to file in the DCGI body
Yeah,
for
sir. Actually, after the preclinical research, if you found a compound is acting good, if you establish the efficacy in the
yeah.
preclinical research, we can
Yeah.
directly with the whole data, we can apply to the DCGS. First, we have to apply to the DCG, like the
Okay,
CDSC,
means
the
there is
regulator.
no requirement of that human ethical permission previously.
No, sir. No need of human ethical committee permission. First, we have to get the CTNOC, as I said, the clinical trial no objection certificate from the regulatory body. Then we have to select the clinical trial sites as well as the ethical committees. Till then, no need of ethical committees.
Okay, okay. Yes, anyone, students, any question? If you want to ask, then
Actually, I could not able to access the chat box here. Like if you send
no, no.
anything else.
Okay, okay. If somebody will ask, then I will.
Sure, sure,
Yeah,
sure.
yeah. Students, any confusion? If you want to ask any question regarding your, yes, if anybody interested in clinical research or clinical trials or looking as a career option in the clinical trials, so they can ask questions if they have any doubt. Yeah. Okay. Okay. Asta ma'am, please give vote of thanks to the sir.
Okay, sir. Good afternoon, everyone. We are privileged that we had with us today Dr. K. Dilip sir, Scientist C, Division of Epidemiology and Communicable Disease, Indian Council of Medical Research, New Delhi. Sir, on his deliberation, explained new drug development process and phases of clinical trials that in detail, that is from sponsors to the study subject. I hope that your easier mode of explanation is easily grasped by our scholars and students, and it will be very fruitful to them. Thank you, sir. I'm also thankful to Dr. Sanjay Gupta, sir, for coordinating this webinar. I also acknowledge the gratitude of Principal and Professor Dr. Chanchaldip Kaur and our Director, sir, Sri M.K. Srivastava, sir, for providing us the platform of VTalk series so that we get enriched by different topics which are going on in research. I'm also thankful to students, the scholars, and the participants who have attended this seminar. Thank you very much, sir. Thank you very much, sir, for connecting us. Thank you, sir.
Thank you, ma'am. Thank you, everyone. Thank you.
Thank you, sir. I hope in the future
Thank-
also we'll get the opportunity to listen to you. It was a
Sure,
really
sure, ma'am.
enriching program. Thank you so much, sir.
Thank you, ma'am. Thank you.